Hello, everyone. Our lesson for today is about transcription. So, in this lesson, you will be introduced to the structure of a gene, the RNA polymerase core enzyme, and the different events in transcription. After this, the molecular details of the transcription initiation and termination will be elaborated. So at the end of this lesson, you will be able to describe the structure of a gene, describe the structure and function of RNA polymerase core enzyme, discuss the events in transcription, identify the different initiation factors in prokaryotes and eukaryotes, and differentiate the termination process in prokaryotes and eukaryotes. But before we formally begin our discussion on transcription, I wanted to give you an exercise first, no, as a review on the basics of the transcription process. So before, in your previous lesson on genetics, you were introduced into the basic concept of transcription. So you were able to transcribe a DNA fragment into an RNA, into a messenger RNA. So you have here a given DNA segment. So I want you to transcribe this DNA segment. So you have here the 3' to 5' strand and a 5' to 3' strand. Okay. So do you have the answer already? Okay. So you are correct. The mRNA produced after transcription of this given DNA segment is 5' AUG, CCG, AAU, GGU, CU, ACG, CG, CAU, AA 3'. So we know that direction of transcription is from 5' to 3'. No, it cannot synthesize from 3' to prime. That's why the strand that must serve as the template is the 3' to 5'. But what you are copying is actually this segment. The 3' to 5' only serves as the template, because if you use this as a template, you will be able to copy this. No? But then this one is our target, no, DNA segment. But we will talk more of that as we go into our discussion. So the question is, how does the cell able to synthesize an RNA molecule? So how, no, is this thing happening? So we will look into the molecular details of this process. So as a definition, transcription is the process wherein genetic information from DNA is transferred into an RNA. It is the first step in the expression of the genetic information stored in the genome. So from gene to gene product, which is frequently a protein, but not all, because RNAs, no, the RN the like ribosomal RNAs are also transcribed, no, from a certain segment in the DNA. But then these ribosomal RNAs are no longer transcribed into a protein. So the end product of the expression of a gene may is frequently a protein, but not always. So it became very rapidly clear in the 50s that DNA could not be directly converted into protein. No? So that is impossible. So they proposed that there had to be an intermediate step. So that unstable product, no, the intermediate between the protein and the DNA, is identified as RNA. RNA is a single-stranded molecule that contains uracil instead of thymine. You already know that one. And it uses ribose sugar instead of deoxyribose, like the one that you shown now in the during your genetics class. So if we take a look again at the central dogma of molecular biology, we know that DNA, no, is transcribed into an RNA in a process called transcription, and RNA is expressed into a protein in a process called translation. Now, in eukaryotic system, now the transcription occurs now in the nucleus and translation occurs in the uh, cytoplasm now of the cell. So we know that the main player now in the transcription process are the RNA because they are the end products. So what are the different classes of RNA? There are three major classes of RNA. So these are called the ribosomal RNA or the rRNA, the transfer RNA or the tRNA, the messenger RNA or the mRNA. So these are the major classes that participate in protein synthesis and are synthesized from DNA by transcription. Now, there are also other forms of RNA like the small nuclear RNA or the snRNA, the small nucleolar RNA or the snRNA, the microRNA or the miRNA, the long non-coding RNA or the linked RNA, and the siRNA, which is involved in RNA interference. Now, we know that RNAs are transcribed from a gene. So what is a gene? How does a gene look like? What are its components? So a gene is a segment or segments of the DNA with a distinct sequence of nucleotides that is transcribed and translated into a protein product that contributes to the expression of a specific phenotype. So a gene, you have here a DNA, the deoxyribonucleic acid, and portions of that are sequences which could be coding for a certain trait, a certain protein. So those DNA that are coding for a certain trait or phenotype are called genes. And in the particular stretch of DNA, you can find so many genes. So for example, now, a specific sequence in the DNA of humans may code for a protein for hemoglobin, which gives red color phenotype to red blood cells. Or some protein could code for melanin, responsible for dark color phenotype of the skin and the hair. So there are different types now of genes. However, now, the protein product of a gene may also be an enzyme responsible for a specific function, such as now the enzyme amylase that breaks down the starch in seeds, resulting in seed germination. So, not all of the time that gene are expressed into traits or phenotype. So they could also be processes are also that are also phenotype or are also products now of gene expression. So if you look into the structure of a gene, no, so you will see that genes of organisms are composed of a promoter region, okay, the RNA coding sequence, or what we call the ORF, or the open reading frame, and the terminator region. So the sequences after the transcription initiation site towards the 3' end of the template strand are called the upstream elements, while those sequences before the transcription termination site towards the 5' end are called the downstream elements. Okay, again, you have the structure of a gene, which is composed of the promoter region, the RNA coding sequence. So this is the one that will be expressed later on into an mRNA. So these sequences here help now in the transcription processes. So you have the ORF or the RNA coding sequence and the terminator region. Those sequences now that are found upstream or before these promoter region are called the upstream elements, while those before the termination sites no, are called the downstream elements. Now, in order to produce the mRNA from the particular gene or from the DNA, the enzyme responsible for that is called the RNA polymerase core enzyme. Okay? So it's almost the same, but it is different from the DNA polymerase. So the DNA polymerase is the one that is involved in DNA replication. So when you when you replicate a DNA to produce more DNA copy, the cells would use a DNA polymerase. But when you express or when you transcribe a DNA into a messenger RNA, the one that does it is the RNA polymerase core enzyme. So the core enzyme has 5 units, which is approximately 400 kilodalton. No? So you have the beta prime. This is the largest subunit, which is encoded by the RPOC gene and contains part of the active center responsible for RNA synthesis. The beta subunit is the second largest subunit. So you have here the beta prime, the blue one, and you have the beta. Okay. This contains the rest of the active center responsible for RNA synthesis. No, so we know that this is an enzyme, so it must have an active site where the catalysis would occur. The alpha are the third largest subunit and is present in 2 copies per molecule of RNA polymerase. So A1 and A2, or alpha 1 and alpha 2, and each contains 2 domains, the alpha N terminal domain and the alpha C terminal domain. So depending on the structure of the protein, so we know that proteins have an N terminal and C terminal, so the amino end and the carboxyl end. So the alpha NTD is required for assembly of RNA polymerase. The alpha CTD also is required for interaction with the promoter. So they're non-sequence specific interactions at most promoters and sequence specific interactions at upstream element containing promoters. Alpha interacts with regulatory factors as well, not the one that regulates transcription of certain genetic elements. This one here, the omega, is this an omega? Okay, so yes, this is an omega. This is the smallest subunit, so it facilitates assembly of RNA polymerase and stabilizes also the RNA polymerase. So RNA polymerase in all living organisms are evolutionary related. So you have here a comparison of the structure of the bacteria, archaea, and eukaryotes. So in bacteria, there is only one type of RNA polymerase, as well as in archaea. But in eukaryotes, there are 3 types of RNA polymerase, and even in plants, there is additional, the 4th and the 5th. So if you notice here, the structure of the archaeal RNA polymerase is more or less the same with the eukaryotic and not with the bacterial. Okay, so archaeal RNA polymerase is similar to eukaryotic RNA polymerase 2, which is the main RNA polymerase responsible for most of the transcription of genes in eukaryotes. Okay, so we already know about the components, the structure of a gene, the main player, which is the RNA polymerase. Okay, now let us talk about the different events in transcription. So how does an mRNA is produced from a gene? So RNA polymerase will proceed through a series of well-defined steps that are grouped into 3 phases: the initiation, the elongation, and the termination. During initiation, the RNA polymerase, together with any initiation factors required, they are the initiation factors are the one that helps in the transcription process, binds to the promoter and the DNA sequence where transcription starts. So it's very important that there is a promoter region in a gene. Otherwise, the transcription or the initiation factors will not be able to locate where to start the transcription process. So once formed, the promoter polymerase complex undergoes structural changes that is required for initiation to proceed. The DNA will then unwind around the point where transcription will start. The base pairs are disrupted, producing a transcription bubble of single-stranded DNA. So the new ribonucleotide is added to the 3' end of the growing strands, since transcription always occurs in 5' to 3' direction. So the one exposed should be the 3 prime so that a 5 prime will be added. Okay, so this is the direction of the synthesis. So you have here an RNA polymerase that synthesizes this. Now, in a particular DNA segment, so you have here the sense strand and the anti-sense strand. So only one DNA strand acts as a template on which the RNA strand is built. So this is what I was telling you earlier during the before we started our discussion. Okay, so the strand is called the template strand or the antisense strand or the non-coding strand. The other strand to where the code being copied is located where the code being copied is located is called the non-template strand or sense strand or coding strand. Because RNA polymerase binds promoters in a different orientation, the same strand is always transcribed from a given promoter. So the choice of promoter determines which stretch of DNA is transcribed and is the most common step at which regulations are imposed. So you have here a given DNA fragment. So you will know immediately if you're given this type of fragment. You will know immediately which a fragment or which strand is to be copied and which will serve as a template, because we know that the direction of the RNA synthesis is from 5 prime to 3 prime. So automatically, the strand that is 3 prime to 5 prime is the template strand or the antisense strand, because this is not the one that is being copied. This strand here is to be copied. That's why it is called the sense strand or the coding strand. So in order for you to copy this, you must follow, so the RNA polymerase must use this as a template. Because if this strand is used as a template, then it will be this strand that will be copied. Okay? So after the transcription, you will now have the mRNA strand that is produced. Okay? And during translation, each of these will correspond to particular amino acid. So we will talk about that in the next lesson. So after initiation is the elongation. So once the RNA polymerase has synthesized a short stretch of RNA, it then shifts into the elongation phase. So during the elongation, the enzyme performs an impressive range of tasks in addition to the catalysis of the mRNA synthesis. So, what happens is that the RNA polymerase will unwind the DNA that is in front of it and reanneals the one that is behind so that they will not be degraded. Okay? It dissociates the growing mRNA chain from the template also as it moves along and performs proofreading functions. So they have so many tasks. No? So it is really a multitasking activity for the RNA polymerase. So you recall that during replication, in contrast, several different enzymes are required to catalyze a similar range of function. And if we recall during the DNA replication process, a helicase is responsible for unwinding the helix. For untangling, you have the topoisomerases, DNA polymerase for the synthesis. And so in here, the RNA polymerase does all the job. Then finally, once the polymerase has finished transcribing the protein coding region of the gene, it must stop and release the mRNA product as well as dissociate from the DNA. So this is now what we call the termination phase of the transcription. In some cells, specific animal characterized sequences signal termination of transcription. In others, it is less clear what triggers the enzyme to stop transcribing and detach from the template. Okay. So you have here we have already discussed the three events: the initiation, the elongation, and the termination. Now during initiation, we talked about the promoter region. No? It must be located by the RNA polymerase so that they will understand where to start the the transcription process. So if we compare prokaryotic and eukaryotic, there's a big difference in their promoter regions. Okay. So again, bacterial promoter regions are located upstream of the open reading frame or the protein coding region of a gene. So the prokaryotic promoter region has a transcribed site or initiation site at -10. -10 because it is already upstream. So the nucleotide here, the very first nucleotide of the open reading frame, the protein coding region, is called the +1. Okay, and those before that are now called the minus the region. So -10, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So at this site here, the -10 and -35, the DNA region, is the transcription start site or the initiation site in prokaryotes. Okay, so it is located 10 nucleotide upstream of the transcription start site. Okay, and it's known as the Privnob-Schaller box. Okay, so that -10 here. So the -35 region is located 35 nucleotides upstream of the transcription start site. So you count to 1, 2, 3, 4, 30, up to 35. And many prokaryotes share common similar sequence at their -35 and -10 region. So these shared sequences are called consensus sequences. At -10 region is the sequence tataat. T-A-T-A-A-T, tataat, -10. And at the sequence -35 is the titigaka. Okay, so you should remember that. Okay. So the strength of a promoter is reflected by the match between its -10 and -35 sequences and the consensus sequences. Now for these regions also, contact between the alpha subunits of the polymerase and the sequence between positions -40 and -60 upstream can contribute also to the promoter recognition and the strength. Okay? So this is for the prokaryotic promoter region. In eukaryotic promoter region, okay, so this is located at -25, okay, and at -80 DNA regions upstream of the transcription start site. These regions also have consensus sequence. At -35 here, the sequence Tata, or what we call the Tata box, is found. While at -80 here is the sequence Kaat, or the Kaat box. So if you notice here, -10 and -35. -10 Tataat, -35 Titigaka. For eukaryotic, -25 Tata box, and -80 Kaat box. Okay? So in prokaryotes also. So we have already discussed about the promoter regions now of the gene. Now what directs or what guides the RNA polymerase to these promoter regions in the particular gene are what we call initiation factors. Okay. So in prokaryotes, the initiation factor is called the sigma factor. So what does it do? The sigma binds to the promoter region that will recognize both the -35 and the -10 regions, forming a close pre-initiation complex. So the sigma provides the helicase activity that unwinds the DNA to form the open complex. So following initiation, sigma is then released. So after, if the job of the initiation factor is already finished because the transcription has already initiated, then it will be released from the holoenzyme only after the first 10 nucleotides of the messenger RNA have been synthesized already. Now, the RNA polymerase containing sigma 70 is the major polymerase species present during exponential growth. But there are also some sigma factors that are involved in special circumstances, like for heat shock response, for nitrogen assimilation, and etc. In eukaryotes, the initiation factor are called the transcription factors, or the TF. Okay, So the core promoter is where the assembly, okay, of the transcription pre-initiation complex, the PIC that includes TF2A transcription factor 2B, 2D, 2E, F, H, and RNA polymerase 2 occurs. So it is at that core promoter. The Tata binding protein is also part of the TF2D that binds the Tata back. So this is the case for eukaryotic organisms. So again, for prokaryotes, the initiation factor are called the sigma factors, while for eukaryotes, they are called transcription factors. Okay. So during termination, again, we know that there are termination sites, no, in a gene. So in prokaryotes, there are two main mechanisms for transcription termination: the row independent and the row dependent termination. So how do they differ? Row independent termination occurs when the termination sequence forms a G-stem loop in the nascent RNA, which causes RNA polymerase to pause. So the stem loop is followed by a stretch of the uridine or uracil residues, which destabilizes, the polymerization process and cause the release of the RNA chain. So this is what we call row independent termination. No? Obviously, there is no row proteins involved. While if in the case of the row dependent termination, it is following the hot pursuit model for row action that suggests that row binds to a nascent RNA chain. So this might require specific sequence or not. So raw then translocate. Okay? The raw translocates along the messenger RNA. The stretch of RNA DNA hybrid in the translation bubble can be directly accessed by raw and cause it to unwind by its helicase activity. Completion of the eukaryotic termination is distinguished by the release of RNA polymerase and raw from the nucleic acid. Okay? So that's how transcription is terminated in prokaryotes. So it could be row dependent or row independent. Now uh in eukaryotes it is different. They cannot just terminate it because there are still so many events that occur. So once transcribed, the eukaryotic RNA has to be processed in various ways before being exported from the nucleus and where it can be translated. So these processing are called the post-transcriptional processes, including the capping of the 5' end of the RNA, the polyadenylation of the 3' end of the RNA, and splicing. So capping is the process that involves the addition of a modified guanine base to the 5' end of the RNA. So specifically, it is a methylated guanine. Specifically, it is a 7-methyl guanosine cap. Okay, 7-methyl guanosine cap that is added to the 5' end of the growing transcript by a 5' to 5' phosphate linkage. So you have here the direction of the base here. Now is the different direction. That's why 5' is exposed. So 5', 5'. So the 5' cap will protect these mRNA. So when we say nascent mRNA, we're talking about the mRNA that has just been transcribed in eukaryote and has not yet undergone post-transcriptional processing. So it's the 5' capping prevents the nascent mRNA from degradation. It promotes splicing and regulates nuclear export. It also assists in ribosome binding during translation. So at the 5' end of the newly transcribed mRNA, you will have the 5' of the 7-methyl guanosine cap. At the 3' end, because why does it need to be protected? Because these are single-stranded nucleic acid molecule, RNA. So it is very prone to degradation. That's why the end of this must be protected. So first is the capping, and the second is what we call the polyadenylation of the 3' end of the mRNA. So this is intimately linked. No, this is how they will terminate the transcription. Because capping can occur once the messenger RNA uh, has already been a few sequences must could be transcribed already. So once polymerase encounters the specific sequences at the end of a gene after being transcribed into RNA, the transfer of the polyadenylation enzyme to the RNA is triggered. So this leads to the occurrence of four events. First is the cleavage of the message, the addition of many adenine residues to its 3' end, degradation of the RNA remaining associated with RNA polymerase by 5' to 3' ribonucleases, and subsequently, the termination of transcription. So the poly A tail protects the mRNA that has just been transcribed from degradation. So it aids in the export of the mature mRNA to the cytoplasm. Finally, it is involved in binding proteins responsible for initiating translation. Okay, so for the protection of the messenger RNA transcribe, they are given the cap, the 7-methylguanosine, and at the end there is polyadenylation. So polyadenylated meaning at the end there is multiple adenine residues that are being added. Okay? And this is important and this is useful because knowing this would make it easier, for scientists to isolate express mRNA, at the so when you study transcriptomics, it will be easier because uh, you will be able to directly capture the mRNA expressed because of this poly A tail. Okay? Now the last process, post-transcriptional process, in eukaryotes is what we call splicing. Okay? So unlike prokaryotes, eukaryotic genes have non-coding sequences. No, unlike in eukaryotes, if they may have, it could be very little. No, so they don't have non-coding regions. All of the regions in a prokaryotic genome are gene coding. In eukaryotes, it's not. No, because there are non-coding sequences that are inserted in between the coding regions of the genes. So these non-coding DNA sequences are called the introns. Now they are inserted in between the coding regions, which are called the exons. That's why the final stage of RNA processing after the termination of transcription is the removal, of the introns. So you have here the cap already and the poly A tail. But this cannot be expressed directly into a protein because there are still non-coding region. So this needs to be removed first. So that's why the process is called splicing. No? This is carried out in a series of reactions catalyzed by the spliceosome, which is a ribonucleotide complex. So we will not talk in detail about the spliceosome, but just to give you an idea of the splicing. So splicing is basically the removal of introns and the joining of the exons together to form the protein coding region. So how does it happen? Okay. So splicing is a two-stage process. First is the formation of the 2' 5' phosphodiester bond between the lariat site, okay, in the intron and the 5' phosphate. And second, the free 3' OH of the 5' exon will generate phosphodiester with 3' exon that releases the intron lariat. Okay, so this is two-stage trans-esterification reaction. So we have here the first transfer of ester linkage and the second transfer of eastern linkage. So the intron lariat is then debranched and degraded. Okay, so take note that the trans-esterification reactions do not require free energy input. So this is how the splicing is done. They remove the introns and uh, they join in the exons together in a two-stage process ng two trans-esterification reaction. Okay, so we have just discussed the molecular details of transcription, starting the different events of transcription, the different components of transcription, the different processes, and the molecular details of the structure of a gene. So in order for you to review for this lesson, now you may want to answer this question. What is transcription? What are the key players in the transcription process? How does translation occur? How does transcription occur? Can you explain the molecular details of each stages? So I hope you will be able to answer this one as you go review this lesson. So if you have a question about the topic, about the lesson that we have discussed, you feel free to contact me through my email address, no? Or through the email address of your instructor, of your lecturer. You can directly key in your question also to the various group chats, or in our case in our Facebook Messenger group chat. You can directly key in your question there. Okay. So thank you very much, and see you again in our next lesson.